My name is Stephanie Moyles, and this evening I'm going to take another path like Jura, but mine is even more veered off the routine hospital path. So I'm actually going to talk to you about the pharmaceutical and medical device industry and my career through both of those actually at this stage. So as I said, my name is Stephanie Moyles, and I actually graduated from medical science here in GMIT in 2010. And after that, well actually before this, like most people at that stage, I hit the road, I flew off to Australia. I had been travelling every summer anyways, instead of staying in the hospital and doing my lab aid work for free. So I couldn't wait to get going. And off I went thinking I'd just walk straight into a hospital there and be a medical scientist and go to the beach every day and be great. But I wasn't long there when I realised that really without placement over and above, your without experience over and above your placement that you would have done as part of the course, especially in a different country, you're not really going to get far. The other thing that I did meet a lot of were questions about whether my qualification was transferable in Australia. So definitely if you are thinking of taking your medical science degree elsewhere, I just make sure that you have all of that kind of your T's crossed and your I's dotted before you head off, so you don't meet any unnecessary questions. So I came back from Australia, and there was HSE embargoes and there was slim pickings. There wasn't much in the private hospital, and there definitely wasn't anything in the public hospital unless you were willing to go in and work for free, and that's not what I was willing to do. So I figured I'd better widen my search, and I thought, okay, well, what do I widen it to? What else can I do? Unfortunately, we didn't have an evening like this when I was here, and you guys really are lucky to be able to get a wide variety of what options are available to you. The only thing that I did have in my head was Mary Grad telling us that when she was qualified and there was a recession that they went off and went into industry and it was all fine because eventually the hospital lads can call in and there was jobs everywhere. And so I thought, okay, yeah, I'll go into this industry, crack for a while and see how I get on. So then I thought, okay, all right, I'll apply for a job, and again, didn't know what I was applying for, and in fact I actually applied for one job. I was about 20 years experience and a PhD short for, but that's just, a qualified person doesn't just mean a person who's qualified, but that's for another day. So I went around with my CV and I got an interview and actually a job pretty quick even, in Baxter in Castlebar in the microbiology lab, and I was there for about a year and a half. And I moved then to Milan, which was Bionish out in Invern, and I was a microbiologist there as well. And the industry lab, be it pharmaceutical or medical device, isn't too dissimilar actually from a medical microbiology lab. I suppose one of the big differences would be your sample type. So you're not looking at feces or urine or bodily fluids. You're going to look at water or products or your environment. So in your pharmaceutical lab especially, you often may have products that are manufactured aseptically. So this means that they're not going to be thermally sterilised. And in cases like this, your environment is particularly important. And by environment, I mean not only the manufacturing environment, but also the personnel that are manufacturing the product. So in this case, if you have an issue with, say, bioburden or surface bioburden or air burden or particulate in your clean room or on one of your personnel that are touching the product, and this gets into a product that's not thermally sterilised, it's a critical significance really and impact. So this needs to be continuously monitored and results assessed and any impact acted upon. So and then from the medical device perspective with microbiology, is some of the sterilisation techniques that we use, for example irradiation, there's actually a direct correlation between the bioburden that's on your product and the particular E being the strength of your electrons and strength of your dose that you're going to give the product. So in cases like that, if there's a change in your bioburden, you can actually no longer have a process that's sterilising. So that's very important to monitor and to keep control of. So I was in the lab in total, I suppose, year and a half, it's about 3.5 years, nearly 4 years, and I was itching to get going again. And so I looked at my options, and I found myself moving not only from pharmaceutical to medical device, but also from a lab-based job to a desk job. And there was a couple of different challenges within that. Obviously, pharmaceutical to medical device is a whole different ballgame. You kind of think of it all as industry, but it's not. They're very different standards, very different auditing bodies, notified bodies. So there's a lot to contend with. But actually what I found to be quite a challenge was to move from a lab-based role to a desk job, and especially because I was moving into validations. And the challenge I found here was because in a lab, usually you've quite daily tasks. So you come in the morning and you know, okay, I need to get this test on and get this test off and get the results through. Whereas when you're in validations, you could have a project that's ongoing for months or even years, or even is never ending. And so to manage your time on that, it's a funny one to get your head around. But it's definitely something that I enjoy, and I'd never go back. Validations is a great area to be in, and you're constantly thinking, and there's a lot to go with it. And there's so many different areas. So it's definitely an area that I would recommend to anyone that would be interested. So first of all, I was in Boston Scientific. I was there for just short of a year, and I've since moved to Medtronic, and I'll be there 2 years in February. So Medtronic, for anyone who doesn't know, we have one site here in Galway. It's the biggest medical device company in the world, especially since acquiring Covidian. There's also a site in Galway, but that's now known as Medtronic. One of the biggest businesses in Medtronic, and what the Galway site is involved in, is cardiovascular disease. And as part of this, we manufacture drug-eluting stents, balloons, stent grafts, inografts, so a lot of different devices. And I won't go through them all now because we'll be here all night. But if you have any questions, if you want to come up to me afterwards, or obviously you can check out the website. I'm not selling any jobs here or anything, but so my current role is I'm a quality engineer in Medtronic, and I specialise in sterilisation, validation and biocompatibility. So essentially my job is to make sure that the product is safe for the patient that it's being either used on or being put into. And what I'm looking at as regards that safety is it sterile? In other words, are you going to put that product into a patient, and especially, most likely that patient is going to be immunocompromised. So you don't want to put in a product that's full of bacteria or, other that's going to go in and cause a pyrogenic reaction or infection. We also want to verify that the product is biocompatible. So in other words, you don't want to put in a stent that's going to be there long term in a patient, but it's actually going to cause them cancer. So you don't want to solve one problem and cause another one while you're at it. So it's very important that we look at these two areas. So typical tasks in these two areas would involve, first of all, validation. So we need to validate a process by which to sterilise the product. We don't validate biocompatibility. So the materials of a product are either biocompatible or they're not. So biocompatibility is more of a yes, it is, and then if there's changes, we assess them. Whereas sterilisation it can be quite an ongoing process, because you can validate a product, but you're validating that product as it is, how it's currently manufactured, and how that sterilisation process currently is. So if there's any changes along the way, you can impact on that, and it may seem minor, but the end result can actually be a major impact on the patient receiving the product. So we do an initial validation, and that validates what the sterilisation process is on a routine basis for a commercial product. And then we reassess and we revalidate that particular process for that particular product on an annual basis, and that's ongoing. And then we also assess if there's any, as I said, any changes can have a major impact. So we assess any changes to the product, and this may be, something significant, like are they changing the metal in the stent, or even as simple as the change in the ink that's in the carton on the outside of the pouch of the primary packaging. And then a huge part now, and it's becoming a bigger and bigger area for us, is audit support. So medical device companies are subject to audits from regulatory agencies and also notified bodies. So we're regularly audited by several different bodies, and of course, microbiology and indeed sterilisation is such a huge, can have such a huge impact on the product and the safety of that product for the patient, that it's always asked. There's always a question from every auditor on either microbiology or sterilisation or both. So definitely we're heavily involved in audit support, and that's never going to change. So some of the technologies that are employed for sterilisation in industry are ethylene oxide, which is a gas. There's also irradiation, for example, gamma, E-beam, and then we also use steam or dry heat. To choose your technology, that's definitely the role of the SME, but it's not singularly the role. So usually at the beginning, at the forefront of a new product development, you'd have some sort of discussion and correlation between the R&D group and the sterilisation group and biocompatibility group to make sure that you're choosing the correct technology. And often it's not a cut and dry, oh yeah, this one ticks all the boxes. It can be, how many boxes can you tick that this is the best technology? So for example, if you had a product that's made composed of materials that are quite likely to degrade or are highly degradable, you definitely would not go for an irradiation technique. And you look then, and similarly, if you had a product that had a high bioburden count, you might prefer to go for a more robust technology and not going to overkill like ethylene oxide. But again, that's kind of more technical there, so we'll leave that at that. So at the moment, my kind of current role is I'm validating 2 new products into Galway. It's not a usual occurrence for someone to be validating 2 products at one time, but I decided to do this, and the reason for this is because we are continuously working to improve processes. Everywhere, no matter where you work, be it public or private, you have resource constraints, you have cost constraints. You're always trying to improve processes, have cost savings. So what I've done is I strategized that I took 2 new products coming in, knew they were coming in within a year of each other, strategized a validation strategy where I can not only validate the 2 products together, but I will also then be able to allow that product team, that business unit, to have the capacity and flexibility to sterilise those products on a commercial routine basis, either together or singly, and have an option of 3 different sterilisation chambers. So that mightn't sound, anything too special, but for us it's a huge deal, and it means instead of creating more and more cycles that we have to, or processes that we have to maintain and revalidate and constantly on an ongoing basis control, we've just reduced it, by half. So definitely, if it's successful, and hopefully it will be, because I want to keep my job, it's something that people will be doing, more and more of. So it's kind of a trial and error at the minute, but it's a good learning opportunity, as I keep being told. So co-sterilising. So if it might sound like, for example, ethylene oxide is processed in a chamber, because ethylene oxide is highly flammable and cannot come in contact with air at all, or else we'll have a huge explosion. So the chambers that we use are in Tullamore, and so what's routinely done is we will put one product into the chamber and that's sterilised on its own. But with this project, we'll actually be able to, the teams and the planners for each of Aptus and atherectomy products will be able to decide, okay, I don't have 12 pallets of product this week for my sterilisation cycle, but you have 6 and I have 6, so let's go together and we'll get a full chamber. So you are, using your full capacity of your chamber, and you're getting the highest benefit from a cost perspective of that cycle. So this, I thought, you think you relay this information to team, but unfortunately sometimes it's hard to push what's the norm, and let people know that just because this isn't the way it's always been done doesn't mean it's not the best way, or that it's not equally as good, if not better, than what's always been done. So so this took a little bit of agreement from the teams, but that again is just another challenging part of your job, and something that it's a good part to have. So what we've been able to do is get a cross-function assessments from design assurance, from design verification to ensure that there will be no impact to functionality, and also biocompatibility is a concern in this kind of a project, to make sure that there will be no impact as a result of co-sterilising. So these are two products that I'm working on, and I just have a short little clip just to give you an idea of some medical device products. So just click into this. So this is Aptus, Helifix, and so as I said, a lot of the, I think I can make this bigger, so a lot of the things that we make in Medtronic and Galway are stents or endografts, and what this does is it actually, I'm going to rewind that because it's actually quite short. What this does is, if we've put an endograft into a patient, unfortunately what can happen, it doesn't happen all the time, but what can happen is you can see migration of that graft, and it's not like it's going from your head to your toe, but even a slight slippage can encourage leaks and cause leaks. So what we can do, as you can see here, I'm just going to bring that back to the start, is we can actually use these anchors. And these anchors can be used either when the endograft is first put into the body, or if the clinician is concerned that there may be migration of an endograft, they can go back in with these anchors. And what you're seeing there actually is the line here is, let's say, the graft, and this is the vessel wall. So they go in, they apply the endo anchor to the end of the guide, and they come in and literally screw it to the inside of your vessel, so it's not going anywhere after that. So that is Aptus. Oh no, I'm going to the wrong one there. And then this is atherectomy. So this is completely different, but nonetheless, I'm going to mute that. So what atherectomy is? So atherectomy is a directional atherectomy device, and what you see here is an artery in a patient's leg that is full of plaque. And suppose in the past you may have had to go in there maybe with a stent or a balloon or something to get that plaque moving. But what we can actually now do is go in with the atherectomy device. You can see here there's like I think it's a bit like a cheese grater. I don't want to put anyone off there doing it now, I'm sorry. But we're going in here collecting some plaque off the artery wall. It's actually got a collection vessel on the back end of it to collect, to retreat out of the vessel, discard the plaque, and then the clinician can decide to go back in if needs be and collect some more. The result being that the patient, it's minimally, I don't know what I call it, minimally invasive, but the patient isn't left with an implant and is now free to look after themselves healthily. And you can see here on the two pictures, so here's before, you can see how narrow the arteries are showing up, and then here how much room they have for blood flow. So it's just a little example of some of the products that are manufactured, and you can imagine how important these are to the patients that are receiving them. And that patient doesn't want to receive such a great product and they get an infection or cancer or a mutation afterwards. You know, it sounds simple, but it's true. So how did GMIT and medical science prepare me? So this is actually something that from time to time I kind of wonder, why did I even need a medical science degree? Because I never really used it. What I mean by that is I never really needed that eligibility for membership of the academy that, we do speak quite a lot about. But when I actually think down a bit and I think, I could have just done this with a science degree or an engineering degree. But when it really comes down to it, I have no doubt in my mind that my ethics are as strong as they are, and that the level of the standard is so high that I expect from my team members and from myself, and that is in no small part due to studying here in GMIT, to the lecturers and to the course in itself. And because really the whole time it was reiterated when I was both here and also when I was on my placement, was that there's a patient at the other end of that blood sample, or the other, whatever sample it may be, and that patient's important. And that sense of responsibility that I gained here is something I've brought with me through every job that I've had, be it pharmaceutical or medical device, and it's something that I hope I never lose. And thankfully in Medtronic, it's mirrored by our one of our mottos is, you touch the product, you touch the patient. So no matter if you're a product builder on the line, or if you're in validations, or if you're senior quality director, you are touching that product in some way, and so you are actually touching that patient. So what you do every day is very important. And also, it's just, even whether you use it or not, it's nice to have a background in medical device medical science, and to know a bit about disease, whether it's for your career or in general in life. It's nice to have. So if I had one piece of advice, if I was able to go back and talk to myself, well, that's tell myself a lot of things, but definitely one thing that I would like to say to myself was to stop worrying about what everyone else was doing and just focus on myself. And I know, we've heard that time since we've been tiny little kids, your mom would say it, or your granny, or someone, but it's really true. And it's something that it's only now when I look back that I realise how I didn't need to be worrying about. I used to get so down when someone else would get like really, really great marks in written exams. Like I decided to take 5 years to do my degree instead of 4, and I get so down and worry, like, what am I going to do? And then when I went into industry, I thought, oh God, am I such a failure? Like, I haven't used my degree, and you get so worried. And I never once realised that actually written exam, they're all finished once you leave college, unless you obviously go on to do further study. And what's been one of my greatest strengths, and the reason for my success, has actually been my character, and the fact that I'm such a hard worker, that how I treat people, how I come to team meetings prepared, how you use initiative, how you want to improve, how you want to challenge yourself and always keep growing. If you're in the same role, or if you move into a new role, or whatever position you're in, that you're always pushing yourself, and that you're never settling, and that you are always willing to help and be a nice person. And it sounds so simple, but when you're in a day-to-day job and you're working for 40, 50, maybe on a bad week, even 60 hours a week, it makes a big difference if someone is actually a nice person and you're able to get on with them. So, I think that was Dr. Seuss. I don't know that anyone can say it better than him, but the truth is that only you are you, and never forget that. So when, if it's now, or if it's in a few years' time when you're out in the real world, just remember that if you're in an interview or you're working, just be yourself. And that, if you're a good person and you're trying hard all the time, that'll get you a lot further than you realise. Okay.
We're all motivated now, actually.
Go and take some more. No.
Has anybody anything that you'd like to ask Stephanie, or any comments, or completely different utilisation of the knowledge that you have? And I do think the foundation that you have does stand to you more than mature as well.
Definitely.
Between the pharmaceutical and the medical devices.
Yeah.
For those that have just finished third year, a lot of QM words being, even they just had their QM assessments the third year.
Yeah.
So validation. So that's a module probably that those, sometimes students go, why do we have to do this? I mean, it's extremely important in the clinical setting as well, but certainly in the medical device and the pharmaceutical, there's no escaping it.
Yeah,
and
exactly.
that language is used, and again, like Siobhan said, those terms are expected that you're
familiar with them.
Nobody's going to sit you down and tell you.
Yeah, I actually remember my first interview in Baxter. One of the questions they asked me was, "What's an SOP?" And I was like, it came out without me even thinking about it, and it was from just hearing these things over and over and over and knowing the basics, knowing those things.
So we're back to that. Is there a lot of repetition in the validation procedure?
Not a bit. No. One product is different, and a product can be one way one day and another way, or process, I should say. Okay. So that's actually one element that I love, because it's constantly changing, and it's, you kind of gain some ground of confidence and think, yes, you know what I'm doing, and then it all changes. And it's always, evolving as well in line with standards, and by that I mean international standards. And what we realise is maybe better practice or goal practice. As I said, Medtronic is the biggest medical device company in the world now, so we see a lot from the FDA that we are audited a lot harsher. Obviously I was in Boston Scientific before this, and I can vouch for that Medtronic are definitely audited with a heavy hand, and the FDA have admitted as well, or not admitted, but highlighted that because Medtronic is so big, they expect us to be gold standard, and because of that, the standard that they're going to audit at is that much higher. So it's a tough environment, but it's one that definitely we like to turn in our favour and say, well, yeah, you can audit us as tough as you want and we'll still do great. You know, so it's just a quality standard for us. Yeah.
Is that okay, Phil?
Yeah.
Can I ask if you miss your lab work?
And not in the slightest. Yeah, no, I don't at all. The only thing I would say though is that while I have no hands-on in the lab anymore, in my protocols that I write, I'm still developing lab methods. So at the minute I'm trying to develop a strategy for the apparatus that we saw, so the endo anchors. So the applier that applies them actually has a battery contained in it. So at the minute I'm trying to develop how I'm going to test that battery in media for 14 days without having a false positive. So I definitely still have my hand in that area of it, which is nice. But I'm just not getting the hand dirty, so it's lovely. Thanks.
Thanks, Stephanie.